Hello class. Now we are going into chapter 2, which goes into the details of human chromosome. And so the question is, why do we need to know about the chromosome? So the reason is, one, it contains DNA, and DNA is the genetic material that is passed on from generation to generations. From since humans came to this happened to this planet. So chromosomes becomes a special body within the cells that carries genetic information. But chromosome is made up of DNA and protein. It is the DNA, the four nucleotides of DNA, that code becomes the language for the transmission of inheritance from one generation to the next generation. Also, when the chromosome is passing from one generation to the next generation, it has to be duplicated or replicated. So unless we understand the chromosomal structure, we won't understand the process of replication, number one. Number two, also, there are many DNA templated processes. For instance, replication, then transcription, which is followed by translation. So DNA serves as a template for two important process for protein synthesis, and second for replication. So replication should happen in the most exact fashion before a cell duplicates. So, so unless we understand the detailed structure of human chromosomes, we cannot interpret the process of replication, transcription, and so on and so forth. Particularly, as you know, there are many diseases that happens because of chromosomal abnormality. It could be a single base mutation. It could be joining of two different chromosomal structures by recombination or translocation. So this is a very important chapter, very good for your fundamental understanding. So let me go to the PowerPoint slides. So I'm sure by now you know this is the book, Essential of Cell Biology, for this coursework. And this is the 4th edition. And so I quickly go into the objectives of this chapter. Number one, to understand the structure of chromosome, to understand how the chromosomes are built. So the building block for chromosomes is a nucleosome. So we understand what is the composition of nucleosome. And one of the major composition of nucleosome is the family of protein. And that protein is histone family of proteins. So we go into that. So I would like to start from very simple and try to connect with the first chapter, which was on cell theory. One of the components of the first chapter is cell theory, when we talk about Robert Brown, who described the structure of nucleus, and the cell theory was given by Schwann, Skeleton, and Virchow. And there are three main tenets of cell theory. One, every organism is made up of cells. Cells need to grow. And third is that when cells are growing, they start from pre-existing cells. So as much as there is an emphasis on the structure and function of cells, there is equal emphasis or more emphasis on division of cells. And when cells are dividing, they have to carry the information. So what is that module that carries the information? So that module is chromosome, right? So from cells, you have nucleus, and within the nucleus is housed chromosome. So subsequently, another theory was proposed by Boveri and Sutton that I also briefly mentioned in chapter 1 about the chromosomal theory. That every dividing cells has to pass chromosome to the next generation. If it doesn't pass, it is not giving the information. So if cells don't receive DNA, they are like host. They don't have any instruction, where to go, what to function, how to look. And then these chromosomes become the basis of inheritance. And On these chromosomes are located genes which could code for protein or may not code for protein. They could have regulatory functions, so on and so forth. So we're starting from cell, then we went to chromosome theory. Now let's quickly look at two different cells that comes from eukaryotic world and from prokaryotic world. So from eukaryotic world, nothing best to give example is, the best example to give rather is human cell. And you can see that human cell is supported by various organelles, including endoplasmic reticulum, ribosome, Golgi apparatus, mitochondria, nucleus, which has chromosome, nucleosome is present, nucleolus is present within the nucleus, lysosome, and they are covered by or boundaried by cell membrane. A totally different cell is bacterial cell, which don't carry nucleus. That's the definition of prokaryotes. Their nucleus is there's no nucleus, so the chromosome is present directly in the cytoplasm, and these DNA coiled or bacterial chromosomes are referred as nucleoid. So now let's focus on human chromosome. So I always remind in every class that a human body or a human cell has 46 chromosomes, but don't get confused that there are 46 different chromosomes. Now there are 23 pairs of chromosomes. That's why we are diploid, right? So most of the body present in our, most of the cells that are present in our body are somatic cells. Minority of the cells are germ cells that give rise to gametes in male and female. So chromosomes again become a center of attraction, whether it is somatic cells or germ cells. So look at the structure of a chromosome that is not duplicated, right? So there's a central piece called as centromere. This centromere is very tight structure. I'll explain to you what do I mean by tight. And then this centromere this centromere, the tight centromere, has two arms, which are called as chromatids, right? It could be a short, it could be a long. Short is referred as P and long arm is referred as Q. Now when the chromosome duplicates, right? So you see here, there's an additional chromatids. The relationship between these chromatids are referred as sister chromatids. So at this stage, when the chromosomes are duplicated, cells are ready to divide. So cells get connected to spindle fibers, and that's through kinetochore. The kinetochore attaches to the centromere. And there are various kinds of chromosomes depending upon the presence of centromere. It could be metacentric, it could be submetacentric, acrocentric, and telocentric. So there are various forms of chromosome. Now, why knowledge of chromosome is important, as I mentioned to you, there could be mutation in the DNA basis. There could be chromosomes could break. There are some studies where it was reported that if you take out the cells from your buccal cavity, generally as people who smoke, their chromosomes are broken. So how to look at these chromosomes? So cytogeneticists, they have evolved some colorful markers to study these chromosomes, right? When these fluorophores were not discovered, scientists used to look under light microscopy. They used to compare the length of chromosomes, because let's say in leukemia, if there is a chromosomal breakage or chromosomal translocation where the size of chromosome is either reduced or increased, it has to be measured. Now, that was a very eye-piercing experiment. But now we have various markers, various stains for various chromosomes. So if you see here, 1 to 23 chromosomes, they color differently. So it's easy to identify, it's easy to measure whether it is normal or abnormal, breakage, translocation, recombination, so on and so forth. So unless we understand the chromosomal structure, how can we understand the disease and normal functioning like replication, transcription, and later translation, where chromosome is not involved in translation, but chromosomes become the starting point, or DNA becomes the starting point for translation. So arrangement of these chromosomes, just a side note, arrangement of chromosomes from 1 to 23 in a diploid cell, this kind of arrangement is called as karyotyping. So from chromosome number 1 to chromosome number 23, the 23rd chromosome is a sex chromosome, XY in males and XX in females. Now, what I want you to understand that when you talk about human DNA, it's true that a DNA is double-stranded DNA made up of nucleotides. But the truth is that there are meters of DNA that is present even in our nucleus that has been packaged. Now, when I say package, what do I mean? Packaging means that it has been shortened with the help of proteins. So chromosome don't look that only DNA is present. In chromosome is a complex of DNA and proteins, where DNA is packaged with the help of protein, where long length of DNA has been packaged and super packaged. Okay? So chromosomes are also banded. Now, second, a very important point that I want to make as a for your concept, and I'm making this, I've put this slide mainly because you have good knowledge of mitosis or meiosis, it doesn't matter. But if you look at whether it's meiosis or mitosis, when you go from prophase to anaphase, or from interphase to anaphase, what do you notice in terms of chromosomal structure? We notice that chromosomes are very thin in the interphase stage, like a woolen ball. And then the length of chromosome shortens and further shortens in metaphase. So what is happening? What is the message I'm giving to you? The message I'm giving to you is chromosome is a dynamic structure. It becomes thin, elongated in interphase, but in prophase, metaphase, and anaphase, it shortens. Now, it shortens, I don't want to go into cell cycle, but it shortens because it helps in chromosomal division. It helps chromosomes to break into equal halves, to move into opposite poles. But the fact is that chromosomes are dynamic structure. They can become very, very thin and long, or they could become very short, like in metaphasic chromosomes. You can see discrete centromere, you could see discrete chromatids under the microscopes, under the microscope. Now, when you look at human chromosome, what do you see here? That there are a lot of fibers. But what are these fibers? These fibers are actually deeply packaged, tightly packaged DNA. And if you zoom in further, you will see that if the DNA unwinds, it becomes a spherical structure. So what you see here, you see in this picture, a normal chromosome with chromatids, and then you see the fibers emerging from, and these fibers then become a beaded structure, a spherical structure, and then you see the DNA. So this is the organization of DNA. And so, as I mentioned to you, packaging of DNA happens. That takes the structure of a chromosome that we see as a chromatid. Okay. Now, the two new terms I am sharing with you here, euchromatin versus heterochromatin. So let me go to the whiteboard. So as I mentioned here, I'm discussing two terminologies, heterochromatin, and second is euchromatin. Now, heterochromatin represents tightly or dense chromo. heterochromatin is dense or tight chromosomal structure, whereas euchromatin is relaxed. And so functionally, no, because of heterochromatin is tight, no transcription can occur on heterochromatin. Why? Because it is very, very tight. But euchromatin is relaxed. This makes the DNA, because of the relaxed nature, DNA is accessible to proteins. Which proteins? Proteins that participates in replication and transcription. And what are those proteins, for instance? In replication, it is DNA polymerase, and in transcription, it is RNA polymerase. So, heterochromatin is a tight structure. Now, what do I mean by the tight structure? So, let's draw a rough diagram. So, let's consider this is a centromere of a chromosome. Right? And this centromere is attached to chromatid. So let's say this is one chromatid, and this is the second chromatid. It's not a perfect diagram. I hope the message goes through. And these chromatin, the relationship between these and these two chromatin is that they are sister chromatids. Within this chromatid is DNA. DNA. Now, this centromere is very tight structure. It doesn't participate in transcription, right? So this is a perfect example for heterochromatin. There are some regions of the chromosome that could express protein, right? Some regions, right? Some regions. So the regions that can express protein, such regions are referred as euchromatin. Right? Now, whole chromosome is not euchromatin. A chromosome could be a patch of heterochromatin, or so a chromosome could be a patch of heterochromatin and euchromatin. Now, this is a chromosome is dynamic. It could be heterochromatin, it could be a euchromatin, right? So, but centromere is primarily the heterochromatic structure, tightly bound. Okay, so now let's go back to the slide. So if you focus on this slide, what you see here, centromere, which is a heterochromatin, which is the central portion of the chromosome, right? And then a chromosome is a mosaic of heterochromatin and euchromatin. So wherever there is heterochromatin, you will see that the chromosome is very tight, right? And what does the functional significance of tightness is? That there will be no protein expression. Compared to euchromatin, right? Compared to euchromatin is relaxed, spread out. And because of this spreading, it helps the machinery of transcription or replication to access the chromosome. So chromosome is a mosaic of heterochromatin and euchromatin. Now let's focus on the end of the chromosome. The end of the chromosome is another very heterochromatic structure. That's telomeres. These telomeres, they protect the end of the chromosomes. Why they protect? Because our chromosomes keep breaking. It could be stress related, it could be age related. So at least presence of heterochromatin means that there is no unique gene, there is no vital gene that are present in this region, that if it is broken, then it will lead to disease. There's a thick, dense chromatic structure here, which is referred as heterochromatin. So what I'm taking you now with this slide is that there is a chromosome, and chromosome, the building block for chromosome, if you zoom in, the building block for chromosome is a beaded structure. Beaded structure. So what do I mean? If we zoom in, then these chromosomes will be like bead. Right? This bead. And these beads are connected by DNA. Right? Now these beads like structure is called as nucleosomes. Right? These bead-like structure. So let me give you an analogy. Let's say a macromolecule protein. It is made up of monomer, amino acid, carbohydrate, glucose, nucleic acid, nucleotide. Likewise, chromosome, they are made up of nucleosomes, right? So let's go back to the slide. So organization is very important. And what is an organization? Is that, so let's say, let's focus on this panel, right? Let's focus on this panel. So you see here a DNA that is 2 nanometer in length, right? Now the same DNA, if you package it, in a beaded-like structure, right? Which I meant nucleus, you can package 11 nanometer of DNA. And then when you super package this beads, then the same space, right, can accommodate 30 nanometer of DNA. So what I mentioned to you earlier in the class, that we have our DNA is meters in length, but how they become adjusted into a smaller spherical nucleus-like structure, which is meters in length, it is because of packaging. So packaging of chromosome is very critical. So you see same length, 2 nanometer, but because of packaging and super packaging, it can accommodate 300 nanometer of DNA or 700 nanometer of DNA, and that's how chromosomes are packaged. Right? So watch this YouTube video. Now we come to what are the proteins which are present in the nucleosome. So nucleosomes, this much is clear, that's a building block of chromosome. Now what are the proteins that make up nucleosome? Right? So nucleosome, a single nucleosome is made up of 147 pairs of nucleotide. Some books can say 150, but generally most of the books, they say 147 to 148. And with this 147 pairs of nucleotide associated proteins, and it's a family of protein that is called as histone proteins. So now what are these histone proteins made up of? Histone proteins are made up of proteins such as histone H1, histone H2A, histone H2B, histone H3, and histone H4. This is a classical structure of a nucleosome. So as I mentioned to you that nucleosomes are beaded structures, right? So this beaded structure is a beaded structure, right? This beaded structure, they are connected by a histone protein called as histone H1. So these are histone so two consecutive nucleosomes, they are connected by histone H1. Now, what are these nucleosomes made up of? So these nucleosomes, remember that this is a octameric structure. What do I mean by octameric structure? Means it is made up of 8 proteins. How does this 8 protein comes from? This 8 proteins come from, there are 2 H2A, there are 2 H2B, 2 histone H3, and 2 histone H4. Okay. So what is the total? 8. So these beads, they are octameric structures. And these octameric structures are made up of 8 proteins, right? Histone, H2A, H2B, H3, and H4, right? 2 of each. And what is histone H1? Histone H1 is the linker histone that connects the 2 nucleosomes. And what is the composition of nucleosome? 147 base pairs of nucleotide and octameric structure. So this octameric means 8 proteins, 2 of H2A, 2 H2B, 2 H3, and 2 H4. Now, let's go back to the slides. So now you can ask me a question that how do I know that this is 147 base pairs of DNA? So this was possible by the help of an enzyme called as micrococcal nuclease. That enzyme is called as a micrococcal nuclease. This micrococcal nuclease is a very unique enzyme, right? So what it does, that it cuts at the junction of histones and DNA. And when then scientists, they analyzed this structure, they found that 147 base pairs of nucleotide and octamer of histone proteins. And what is the dimension of one histone? The diameter is 11 nanometers. Right? So look at this slide. This is what I meant. So the crystal structure of nucleosome has also been solved. So this is how a crystal structure of real nucleosome looks like. Now, remember I mentioned to you that chromosome is a dynamic structure that could be existing as a euchromatin, which is a transcriptional friendly zone, or heterochromatin, which is transcriptionally unfriendly zone. It may not permit transcription. So there must be some factors that facilitate transition between euchromatin and heterochromatin. And one of those factors is called as remodeling complex. Remodeling complex. This remodeling complex is an enzyme or a protein, battery of protein, ATP-dependent chromatin remodeling complex. I repeat, ATP-dependent remodeling complex. This means it needs ATP, high energy. And this energy in this protein helps heterochromatin to transition to euchromatin. So heterochromatin in this figure is been shown called as condensed, means tightly packed. In presence of remodeling complex, the chromatin becomes relaxed. And when it is relaxed, it means it facilitates transcription. And when it is dense, it is not transcription friendly. It is inactive, transcriptionally inactive. Right? So ATP remodeling complex, in the presence of ATP, it helps to convert condensed to decondensed chromatin. So now there are other factors that helps the chromosomes to transit between heterochromatin and euchromatin. And so I will go to the slide share. So I mentioned to you one of the factors, one of the proteins that helps to convert heterochromatin to euchromatin is ATP remodeling complex. The second, so this is first. The second is referred as, which converts heterochromatin to euchromatin or euchromatin to heterochromatin. It is called as epigenetic modifications. Now, what do I mean by this is that so I will not go into the detail, but histone acetylation. Where does acetylation occurs? On amino acid lysine. Okay. And this occurs on the side chain of lysine. So when acetylation of lysine occurs on histone, this gives rise to U. Now, lysine is a very interesting amino acid, right? Why I say that? Because lysine can also undergo methylation. What is methylation? Is on the side chains of lysine, methyl group is added, right? On the side chain of methyl. Now it depends where it occurs, right? If the methylation occurs on human histone H3 on lysine 4, so each protein is made up of amino acid, right? So the lysine number 4, if methylation occurs, it causes gene activation. just like acetylation. So euchromatin is associated with gene activation, transcription. The second situation is that if lysine number 27 on human histone H3 gets methylated, it leads to gene silencing, means heterochromatin formation. So gene activation, gene repression are opposite term. Gene activation means mRNA translation will take place. Gene repression or gene silencing means no mRNA or no protein synthesis could be taken. Now these are very important regulatory mechanisms which are present in our cells. Because remember, the logic is that what I want you to understand, that our journey of our life, it starts with a single cell, right? Agree? So journey of our life starts from a single cell, which is called a zygote. Now this zygote branches out, right? Branches out into various organs, brain, kidney, lungs, liver, digestive canal. How? So all these cells, all the cells in the body, they carry the same DNA. But what is the mechanism by which they're distributed into tissue-specific organs? So brain is generally rich in neurons. The job of the brain is to assess the pain, memory, learning, right? The job of liver is digestive enzyme or oxidative mechanism. Pancreas, insulin, thyroid hormone to regulate iodine, kidney filtration. All these organs, different cells emerge from zygote. So zygote is having some inbuilt programming, right? So where does this differentiation comes from? So this differentiation comes from, one of the factor is epigenetic modification, the changes in the structure of chromosomes, the changes of into dynamism of heterochromatin and euchromatin. It's possible that one gene which is good for memory in brain is euchromatin, but in kidney it's not needed. The job of kidney is not to memorize. So there, that memory specific gene could be part of heterochromatin, not needed. So there's differential regulation of genes and chromosomal landscape, chromosomal structure. They play a very huge role in regulation of cellular processes. So with this, we finish chapter number 2. Thank you.